Ken is an associate professor of molecular and comparative pathobiology. And Ken today is going to talk about the body's postal system, which are called extracellular vesicles.
Thanks so much, James, and thanks to everybody for attending today. I'm very excited to share with you some of the work that is coming out of my lab right now, and I'm going to pull up my screen. Hope you can all see this. It'll just take 1 second for me to get into the right one here. So I want to tell you about the body's postal system, and you might notice that my background here is that of a post office. This post office is actually quite an antique post office. It's from Texas. There's a little general store here. And the reason that I chose this antique post office is that I feel like we're in a very exciting time right now in the field that I'm about to describe to you. It might be a little bit of the Wild West too, but we are making progress, and there's a lot of discovery to be made and a lot of contributions, I think, to precision medicine. But first of all, I want to give you maybe a little bit of a public service announcement. As you may know, this year is a little bit unusual, and we're coming towards the time where we all have to pay our taxes, which normally would have been in the past. And as I understand it, this year's tax deadline is on Blur's Day, on April the 15th of this great year 2020. And there are a few things that we have to do when we pay our taxes. We've got to have an envelope here, a package basically that can be delivered. We have to have a stamp, some means of getting it to its destination, and of course we want to have something inside. So just to remind you, you have to tell the federal government who you are. You have to make sure that it gets to the federal government, your tax return. You have to have the stamp there to make sure that that happens, and you don't want to forget to include the check, your payment. This is basically the payload. So the Postal Service is going to be handling a lot of these tax returns in a little while. But does our body have some similar system where we can address things perhaps between different parts of the body? So in the multicellular organism, I want to pose the questions of what packages could possibly be traced back to a cell or an organ of origin. What packages could target then specific places, going to specific addresses? Can these packages negotiate biological barriers, like the barrier between our brain and our blood, for example, between our gut and the blood? And can these packages actually deliver anything that would be meaningful? So I would like to pose to you that the answer to all these questions is the extracellular vesicles. So these are cellular packages. And I want to emphasize, that our body does need to communicate. The parts of our body need to communicate. So my brain sometimes wants to know what's happening in my liver, and the liver might want to know what's happening in the brain. And I think all parts of the body want to know what's happening in the gut, because that's where we get our nutrition from, our food. So that's dinner. So extracellular vesicles, what are these things? So extracellular, the word is obvious. That means that it's outside of the cell. And a vesicle is kind of like a soap bubble. So if you make some soap bubbles or blow some, kids' bubbles, you know what an extracellular vesicle is basically like, except that the extracellular vesicles that I'm talking about have contents in them. And so displayed on this slide are several cell types. So we have some mammalian cells at the top, some bacteria and parasites at the bottom, and everything here is color coded. So as you can see, these little particles, these little packages, the EVs are coming from the cells, maybe from an internal compartment, maybe from the surface of the cell. And they have many, many different names. So over the years, there have been scores of names that have been given to these particles, but altogether we can call them extracellular vesicles. So they are quite diverse, and I want to mention this because that means that there are a lot of different kinds of packages. So depending on what you want to deliver, what kind of a message needs to be delivered in the body, you might have a different particle. Here are some EM images. So these are electron micrograph images. We're going to be seeing a lot more beautiful EM, I think, from Cynthia Wohlberger a little bit later. But here what we see are some extracellular vesicles that my lab has purified and then imaged by electron microscopy. So these things generally are quite small. Now, there are some that are big, but they're generally quite small. They might be about a millionth the size of the cell of origin. And they can have diverse morphology. So in this image, you can see some that almost look like footballs. So up here in the upper right, there's some that actually have a few membranes inside them, but for the most part, they are these spherical particles that are bound by a lipid bilayer and have contents of the cell. And I think we're going to hear more about envelope viruses like coronavirus later today as well. And I would say that envelope viruses themselves are EVs because they are being produced by the cell using the same biogenesis pathways. And something that I like to use when I tell my son about EVs is that they're basically like pokeballs. So if you've ever played the game Pokemon Go, or really any Pokemon game, you will know about these Pokeballs that are these balls, and you can put little pocket monsters inside them, and you can use them to play a game. So there are different kinds of EVs, just like there are different kinds of Pokeballs. And somehow Nintendo actually named one of these the EV. So this is my favorite one. The EV has been known for a very long time. We probably had the first inklings of it back in the 1930s or so. But I think the first time that the word extracellular vesicle was used in a paper, this was actually not a multicellular organism, but a little golden algae called Ochromonas. And electron microscopists could see that there were extracellular vesicles budding from the surface of this organism. And it was just 2 years later when people discovered that there were extracellular vesicles being made in the thyroid of bats that were reawakening from hibernation. And so this showed us that the effects of extracellular vesicles, the association with endocrine processes, was really important. So what is my lab trying to do right now? We work mostly, not entirely, but mostly on brain diseases like Alzheimer's disease. And so I'm showing you an illustration here that I hope will help to communicate how EVs can be involved in precision medicine. So here we see 4 different cell types in the brain. Of course, the neuron is one that everybody knows about, and they're depicted in blue here. But there are other cells in the brain too, some of them even more abundant than neurons, and they're depicted in yellow, in green or gray, and in red. And as you can see, for each of these little tiny spheres that are coming off of those cells are EVs that have come from the cells. And so it's kind of like a color coding. Now, are these really colors? No, but we can label them with colors, much like we saw in Anthony Rosen's presentation. The filters that we can use, the fluorescence labels that we can use for these things are really important. And interestingly, the extracellular vesicles that are found in the brain can also get outside of the brain. They can get into the blood, they can get into the cerebrospinal fluid, even the saliva. So we can track these things in different parts of the body by using surface markers. And so I'm showing here one of the surface markers that we might use for a particular cell type in the brain. Now, when these extracellular vesicles get out of the brain, we can do a lot to understand what is happening in the brain. And here's a study that we did recently with Alzheimer's disease. And we actually decided to look at the vesicles that were in the brain. Before we even look outside the brain, let's find out what's actually at the source. And if we did some studies, some proteomic studies, so looking at the proteins that are found in the brain, we could fairly easily distinguish Alzheimer's disease from control. And what you see here in these 3 boxes is basically some bioinformatics that we tried to use to separate our control brains from the Alzheimer's disease brains. And as you can see, we did a pretty good job of it by just using the tissue itself. Now, if we took our EVs, our cellular packages, and purified them partially from the brain, we still had good separation, but not perfect. But when we took those EVs and made them very pure so that we were just looking at those individual particles, we could completely separate the Alzheimer's disease from the control. And this is important because now what we can do is we can look for keys. So I've been talking about colors. We can think of the different kinds of EVs as having colors, or we could think of them as having these little keys on their surface, keys that betray their origin. And so I'm not going to go into the detail of what each of these is, but let me just say that we did find multiple keys that could be used down the road to assess what is happening in the brain just by taking a blood sample. So not only can we use EVs to understand what's happening in the brain, we can also potentially use them to target tissues. So if we can put something on the surface of one of these little cellular bubbles, we could target it to a particular organ or a particular cell. And this is like a lock and a key. So this little key on the surface of the EV is going to allow it to attach to and maybe be taken up by a particular cell. And I just give a few examples here of the brain as a tissue, a tumor tissue, or even individual cell type like a CD4 positive T cell. So how do we engineer these things? We can put different molecules onto EVs or onto the cells that they come from. We can load EVs with drugs that are able to do something in the tissue that we target. So so this is where we are today. I think it's a very exciting time. We might be more in the Pony Express era right now than in the Space Force era, but we're moving forward rapidly. And I just wanted to let you know there's an international society known as the International Society for Extracellular Vesicles. We have about 1,500 members, and we're having our big virtual meeting next month, July 20 to 22. If anybody's interested in covering this or learning more about EVs and how they can be used in precision medicine, please contact me. I'd be happy to get you a pass to be able to attend this. And so thank you so much for your attention. I just want to give the credit for the work that we're doing to our so-called postal workers and the funding that we receive. So we have funding from multiple NIH agencies and also from some private sources. And finally, I just want to note, and I think that James Berger had mentioned this too. We can't do precision medicine without diversity and inclusion. And so I just think that it's extremely important to remember that precision or personalized, that means people. And so the people who are doing the work as well as the patients need to be considered here. So thank you all for your attention, and I'm happy to take any questions.
I can, thank you. Great. So David Fryer asks, are EVs common to all mammals? Are they also found in other multicellular non-mammalian species like plants or fungi?
Yes, that's I like that question because it lets me talk a little bit more about that diversity of EVs. And indeed, we have found EVs in everything that we have studied. So all multicellular organisms we think have EVs, and even single-celled organisms, as I showed with that golden algae. They also make EVs. So this communication system, it seems to be found in all multicellular organisms.
Okay, well, due to time, we'll have to move on, but thank you again very much.
Thank you.